Okay, it's going in this
According to the customs at this university, we open this academic session with the words Spiritus Sancti Gratia Illuminet Sensus et Corda Nostra. Please be seated. Welcome to all of you, in particular also welcome to Professor Battaglia. And welcome to all the, especially also to all the professors from outside this university, and of course to those inside the university. We will hear the inaugural lecture of Professor Francesco Battaglia. Professor Battaglia studied at the University of Rome, La Sapienza, and obtained his master's in 1994. In 1998, he obtained his PhD, cum laude, at CISA in Trieste. After that, he went for a postdoc to the University of Arizona, and then became an associate researcher at CNRS at Collège de France. And then he came to the Netherlands, which was probably why he, in the end, maybe also landed here. First as assistant professor at the University of Amsterdam from 2006 to 2012. the same time, 2011 and 2013, he was also a part-time visiting scientist at the Neuroelectronics Research of Flanders in Leuven. And from 2013 on, he became associate professor at the Donders Center for Neuroscience at the Faculty of Science of Radboud University, so our university, until 2020, when he was appointed as a full professor in our Faculty of Science. Notably, Professor Battaglia also is a recipient of an ERC Advanced Grant in 2019 and leader of the large Zwaartekracht Consortium since last year, which are very prestigious positions of academic stature, but also giving large possibilities for Professor Battaglia to imprint his knowledge and skills on the field of neuroscience. So I can't wait to hear what he has to say in the inaugural lecture. So please, Professor Battaglia, the floor is yours.
Thank you. So, well, dear Rector, friends, family, colleagues, well, first off, thank you for being here. So this is, of course, a very belated inaugural lecture. So happened that, as Simon just said, I was nominated for professor in February 2020, and clearly wasn't possible to have such a festive event for a long time. I think it's still worth doing this, even after 3 years. And it's not just a self-celebration, but to get the chance to give you a bit the bigger picture of my work. It's not just as an academic presentation where you have to stay focused on a precise topic, but to discuss a little bit with you the overall ideas and give a sense of where we are with our group and where we would like to go in coming years. So what I will do today is tell in brief the story of my journey in science. So for several reasons. First off, because science is not just about ideas and data, but it's about the people that makes it, encounters and long-lasting friendships and collaborations and sometimes random events. Second, many of you are not neuroscientists by trade, and while they may find the scientific part a little boring, maybe they will still find entertaining to know the life side of it. And third, because it's actually a good way to connect some scientific ideas, how they evolved in the 3 decades that I've worked in research and where they may go next. While each life path is unique, I think my career path is actually quite representative of what neuroscience is and has become. And so perhaps the story has some value beyond what my closest friends may find interesting. So I started as a physics student in Rome in the late 80s, early 90s. So the time and place actually matter here. So we were in the heydays of the complexity science, so words like chaos, self-organization, emergent properties, and so on were starting to be the buzz in a number of different fields, so from physics to biology to chemistry to social science. So that was the moment, and the place was the physics department of the University of Rome, and it has to be mentioned because it was quite a special place. So since the time Enrico Fermi founded it, it still is a powerhouse with a massive international reputation. Not that I chose it for that reason. As a young and stupid 18-year-old, I just happened by an introductory lecture, found it cool, and decided to enroll. So very much by chance, I had no idea. So in particular, Rome was and still is a hotspot for statistical physics, with top researchers being at the very forefront of science and getting Nobel Prize-worthy results. In fact, Giorgio Parisi did get the Nobel Prize in 2021. So statistical physics introduces a key idea for understanding the brain and living systems in general and the problems I want to talk about here. That of collective emerging properties. So think of water boiling. Water is made of molecules which can stay together in many different ways, as a liquid, as a vapor, as ice, but the constituents, so the single molecules, stay the same all along. So the overall state of water as a matter depends on the interaction between single molecules, but there is essentially no way to start from those single interactions and find out at which temperature water boils, for example. So, and here, so you see boiling water, and you see activity. So this is half of a mouse brain, and so yeah, and here you see what the activity looks like, and actually you see something that really resembles bubbles. Okay, so yeah, human brain, very big, smaller mouse brain, which is what we mostly look at, and so that's it. So there are emergent properties that depend on the behavior of many units together and cannot be easily reconducted to the behavior of single units. So this becomes most evident when we look at phase transitions. That is the very moment in which water boils, or a magnet acquires magnetization, and so on. So two properties of critical systems, that is, system just at the phase transition, are relevant for what I say later. The first is information can travel very far and very quickly in a critical system. Technically speaking, the correlation length diverges. So even though single molecules or single starlings in a swarm, which actually has been shown obey very similar mathematical equations, they only interact with their neighbors. The effect of a fluctuation somewhere can be felt at a distance. Actually, you can see it here. Sometimes one starling shifts course, and you see that immediately the whole swarm follows. So that's, and it's very similar for matter at a phase transition. So it is as critical system can amplify and propagate information. Second, critical systems tend to follow a small range of stereotype behaviors. They don't care about the details of their microscopic constituents. So because of this property called universality, the link between the microscopic and the macroscopic worlds becomes a bit blurry. So which stereotype behavior or universality class the system follows depends to a large extent from the symmetries of the system. So symmetry is a key concept in physics. We could almost say it is what makes nature amenable to mathematical analysis. So here, hence the typical jokes about physics wanting spherical cows. So going back to my years in Rome, so the biggest deal in that physics department was the statistical physics of disordered systems. So here we move away from symmetry, for example, a perfectly ordered and symmetric crystal, and so for example, in a magnet, so where all the basic elements all interact with each other in an orderly fashion. In a disordered systems, some constituents attract each other, some repel each other with no pattern. So this situation is interesting for us because it's much closer to systems that seems beforehand unattackable by mathematical methods, physical systems, but also biological systems, social network, computing problems, machine learning, and so on. So the brain. The brain is a tangle of connected neurons, some exciting other neurons, some inhibiting. There are some hints at regularity, but mostly it is a tangled mess, random looking at first glance. A typical example of the disordered system is glass. Glass is different from a crystal because the molecules in it are not arranged in an orderly way. So you may remember that when a physical system has an energy function, it will tend to rest in a state where energy is minimal. So in a glass, getting to the minimum, so the dynamics of the system is actually extremely slow. You might have heard that if you took your favorite wine glass or bottle and waited for long enough, which is probably centuries, you would find it splattered on the table just as though it was made of extra thick honey, which in a way it is. So what you have now is a system with much, much longer timescales than its microscopic components. So this property is extremely interesting from a brain point of view. So our brain is made of pieces, cells, synapses, etc., that have a turnaround of hours or days, yet is capable of maintaining certain states that we call memories for years or decades. Where do those long time scales come from? Turns out that glassy dynamics, which is not the way the brain does memory, which actually is something that people believe for a short while. But it's another example of how collective properties can be autonomous from the microscopic way. So Giorgio Parisi, already in the late 70s, was able to mathematically describe spin glasses, magnetic systems with close resemblance to actual glasses, with a tour de force of abstraction and ingenuity, for which he was awarded a Nobel Prize in 2021. Many of the other pioneers in the field either worked in Rome or visited frequently. And as a student, I was, of course, very excited about this, and I took essentially as many courses in that field as I could. So when it was time for me to find a master's thesis, so my plan was to go ask Parisi, who was obviously the first choice, where he was very willing to take me. So I never got there, because out of curiosity, I went knocking the door of another professor that I heard about but never seen before, and that ended up determining my scientific career. So that was Daniel Amit. So Amit was an Israeli physicist and also a pioneer in statistical mechanics who wrote the statistical field theory textbook that I used for some course. So his legendary reputation was also due to the fact that he left Israel as a dissident, having refused to serve in the Israeli army and even having been jailed for that. But at that point, he wasn't teaching a course, so students didn't even know him. So what Amit was mostly famous for was the solution of the Hopfield model. So one of the early and still one of the most important neural network models. The Hopfield model was something like a spin glass, but it's also a model of memory in the brain. So it could give you a rough idea of how brain stores and retrieves information. So to solve this model, Amit uses techniques similar to Parisi's solution of the spin glass. And he did find that under certain conditions, the Hopfield model did show glassy dynamics. However, those conditions didn't match anything that may happen in the brain, so that part of the analogy dropped. So my first interaction with Amit lasted for about 5 minutes. So after 22 years old, extremely intimidated me, looking more as like an 18-year-old, I was told, asked if there were possibilities for a thesis. He replied in his perfect Italian with a distinctly Israeli Polish accent, "Oh, you want a thesis? I give you a thesis." I replied, "Okay, you don't want to know about my grades and so on. Oh, if you want to tell me, it means that they are good. Just come tomorrow, I will tell you what you will do." Okay. So I knew of Amit's Hopfield papers, which I could mostly read based on the courses I took, and I thought I was going to do something with statistical mechanics of more or less that sort. Instead, I was assigned a computational neuroscience project, which is something closer to modeling biological neural networks. So I knew zero biology. I'm not even sure if I had heard the word neuroscience at that point. In any case, I took the next 18 months to read as much as possible about the brain, mathematical and biological papers, which I would find increasingly fascinating. So for my PhD, I ended up in Trieste with Alessandro Treves, a former Amit student. Alessandro was very young, and I was his second PhD student. And the physics approach was quite similar to what I was accustomed to at that point, but with a key difference. So Alessandro was and still is a neuroscientist more than a physicist, and with him I started learning about neuroscience experiment and data. So Alessandro just finished formulating a model of the hippocampus. So here you see a beautiful drawing of the hippocampus. So I don't have an hippocampus tattoo yet, but actually on the back of this toga there is a drawing from Ramon y Cajal of the hippocampus. If you want, I can show you later. His model was endowed with information about structure, about the different subcomponents of the hippocampus, the connections, and their architecture and so on. So the structure is by and large what biological systems are about. So learning about structure is difficult for a physicist, as it's not enough to learn some key equations and use mathematical muscle to derive all the rest. You need to study and learn by heart a lot of information and slowly come to a synthesis. So I wasn't in a world of spherical cows anymore, and that made everything a lot harder. So I got a challenging thesis project. A major known fact about the hippocampus is that it can encode space. You can see neurons in the hippocampus, the so-called place cells, activating when the animal was in a specific location in space. So altogether, those neurons would constitute a map of the environment, or as it has been more recently been put, the brain's GPS. So clever experiments showed that the hippocampus can remember maps for a number of different environments, and this could be modeled by taking inspiration from the Hopfield model. So my task was now to solve the model analytically, and that ended up being very difficult. So I could in part follow the Parisi and Amit solutions for the use of the so-called replica method, but in the Hopfield model, memory aren't just random strings of zeros and ones with no correlation with one another. So in that case, radically simplifies calculations, but here I had to deal with maps, with some neurons sitting close to each other and some further away. So that means there was structure, there was correlation, and that made everything a lot harder. So I had to develop some new technical tools for that, and that took about a year and a half and was the centerpiece of my PhD. So the result was that neural networks storing maps were almost as efficient in storing information as the Hopfield model, which was quite important for the theory of the hippocampus. So at this point, I was knee-deep in the neurophysiological hippocampal literature, but I was still a theoretician. So that changed with another fortuitous encounter. So Katalin Gotthard came to give a talk in Trieste. So Kati was, at that point, in between a PhD and a postdoc with Bruce McNaughton. So Bruce's lab in Tucson, Arizona was the leading lab in recording from many neurons at the same time. What you really needed to track the dynamic of neural systems and understand what information they carry. A few years later, Bruce had introduced the tetrode array, the innovation that changed the way we do electrophysiology, because it made possible for the first time to record from about 100 neurons in a freely moving animal. So Kati was a terrific speaker, and I should say she only got better with time. She showed that they could reconstruct the rat's position as it was tracked by the hippocampus, another technique that was pioneered in Bruce's lab and is now a cornerstone of neuroscience research. So her talk blew my socks off, and that work looked a lot more exciting than calculations with the replica method. So I started thinking that I wanted a piece of that action. So Kati and Alessandro conspired to have me invited for a 3-month visit to Tucson as the final act of my PhD. And I went there and found myself catapulted in a completely different world. So Bruce and Carol Barnes, his then wife lab, was at that time mostly busy with a project to launch rats in the space shuttle to do neurophysiological recording in zero gravity. That required major technical advances and also having to do with NASA logistics, which would be a euphemism to call intimidating complex. So the place was a madhouse with daily panic situation and sometimes quite incensed exchange. And of course, I loved it. So I spent 3 years with my head in the computer analyzing data. After those 3 months, everyone seemed happy, and Bruce offered me to come back for a postdoc. So I came back 1 year later, and this time to do experiments myself. So I hadn't seen an experimental animal before, so I had everything to learn. In typical Bruce's style, being a total beginner, I was put to work on a completely new recording system, the biggest they had at that point, with 144 independently movable electrodes. So this was a team effort, so it wasn't just me, but it was a tech-intensive project. And while I didn't know rats, I knew computer, which actually was useful. So that postdoc set the stage for what I've been doing ever since, or most of it. So we were interested in sleep. And Bruce and colleagues have discovered that patterns of activations of place cells, so these little sequences of cells activating here, that you see during an active experience in a rat or a mouse are actually spontaneously replayed during sleep. And why does that happen? So there are spontaneous activity of fluctuations of activity in the hippocampus that we call sharp waves. And in a sharp wave, that excitation propagates in feedback circuits that look a bit like the Hopfield model. And they would produce this information retrieval or replay. So this was an extremely important discovery which linked the two main ways we think of the hippocampus. On one hand, place cells. On the other, its role in memory, of which I haven't talked about yet. So that story begins with the finding back in the 1950s that a patient, Henry Molaison, then known as HM, after losing both hippocampi, you have two, one per side, to a very primitive surgical approach to intractable epilepsy, could not form new memories anymore. He would remember in a relatively normal ways events from his childhood and had relatively normal cognitive abilities, yet he would not recognize you if you showed up 5 minutes later after you extensively chatted with him and then left. So until these last moments, he wouldn't actually recognize the psychologists like Brenda Milner and then Sue Corkin, who worked with him for decades. So the theoretical idea that this suggested that there were in fact two memory systems, so the hippocampus, that would be mostly responsible for the initial storage of memory, and then the rest of the brain, maybe the cerebral cortex, or what we have all around, that would be for more permanent storage. And a host of data from human and animal experiments supported that. So with time, memory would become less and less dependent from the hippocampus, or as we say in neuroscience, they consolidated. Memory consolidation implies that there is some interaction between hippocampus and cortex after the fact or offline. And the existence of replay would suggest that at least some of it would take place during sleep. So we knew that the hippocampus could replay, and we set out to find where the cortex would listen to that replay. So what I did was then to look at how cortex reacts to the sharp waves, so to these bursts from the hippocampus, which is when most replay occurs. So I found two things. First, that there was an increase in cortical activity during hippocampal sharp waves, small but sufficient to support information transfer. And second, that sharp waves are, to some extent, synchronized with the activity oscillation that the cortex produces during a phase of sleep that we call non-REM. So this was important for two reasons. First, because it suggested that the interaction was two-way, not just from the hippocampus to the cortex, but also the other way around. And second, because the so-called slow wave could act as a powerful amplifier of the hippocampal signal. So our work was first real demonstration of interaction between hippocampus and cortex, and one of the first analysis of interactions between different brain areas. But mostly for technical reasons, we didn't manage to find replay in the cortex, and that came in my next step. So from the desert in Arizona, I went for a complete change of scenery to work at the Collège de France in a beautiful building in the heart of Paris. I also changed approach somewhat. Instead of looking at the cortex as a whole, I concentrated on one area in the cortex, the prefrontal cortex, that has a special link with the hippocampus. At that time, there was a wave of studies looking at interaction between hippocampus and prefrontal, and was lucky enough to catch the beginning of that wave. I was even lucky to work in the team of Sidney Wiener with some fantastic trainees, in particular Adrien Perrache and Karim Benchenan, who went on to become prominent professors themselves. So with Adrien, we developed mathematical methods by which we could actually find the time course of replay, and we looked at replay during the hippocampus in the prefrontal cortex and looking at hippocampal sharp waves, and we found what we were looking for. Replay in the prefrontal cortex is strongly increased when there is this burst in the hippocampus. So we could conclude that hippocampal and cortical replays were connected and could act as the interaction mechanism postulated by the theory of memory consolidation. We also got a surprise, the time course of replay, so its amplitude followed a power law. So here, actually, this is a plot in a log-log scale, and each time you see something on a straight line in this style of plot, it means that's something called the power law. And this is the time of behavior that is typical of systems at a phase transition. For example, the size distribution of bubbles of magnetized material when you cool the magnet to the critical temperature. So criticality in the neural system has been a very active field of studies for about 20 years now. And what we did was to link it to something that had at least an information processing flavor. So power laws were only observed when we looked at the groups of cells that were involved in the replay. So it wasn't just a general population of those neurons. So to continue that story, I have to fast forward 10 years to my Nijmegen years, and then to the work of two great PhD students, Rafael Pedrosa and Morgano Dran, and of Federico Stella, now assistant professor at Donders, and the mind behind many of the theoretical things we are doing. So this idea of criticality stuck with me, but I thought I was not going to understand much by just recording a few hundreds of neurons out of hundreds of thousands in one just small brain area. In fact, I thought where a critical behavior might become important is not just in one single brain area, but when you consider the brain as a whole. So it could be something that connects the neuron in the back of your brain that immediately process visual information, and neurons in the front that will tell you where you will move next. And there you need an effect like this flock of starlings, right? So that amplifies that communication in this web of connections we have in the brain. So that looks like a phase transition, and this kind of things are like fingerprints of phase transitions. So we changed technique a bit, and we used something called voltage imaging. So neurotechnology has advanced a lot, and one thing we can do is, which is a major breakthrough, is that we can use fluorescent molecules that translate the electrical activity of neurons into light that we can see with a fancy microscope. So we set that up with collaborators Majid Mohajerani and Thomas Knopfel, who are luminaries in the field, and started looking at how the cortex as a whole this time and the hippocampus would interact with each other during sleep. So we saw bubbles of activity, and some small, some large, and the size of these events also follows these famous power laws, as depicted here, but now brain-wide. This was known, but to some extent, but not so much. But now, once again, structure gets in the way, and structure is important. So connections between areas are not random, but they follow patterns. So that means you don't observe random subsets of the brain activating together, but very precise subsets that we call functional networks, which is something, a term that originates from the human neuroimaging field. But here, I mean, we cannot look at the human brain because you need transgenic animals and chemicals and other stuff you cannot do with the subject, with a human subject, but we can follow the activity of this functional network with excellent temporal resolution. So we looked at the bubbles, and the bubbles followed very precise shapes. And these shapes followed anatomical connections. And in particular, I'm interested in this shape here, because this looks, is what is known from the human literature as the default mode network. This is a network of structure that is mostly active when you do nothing. It actually gets quieter when you start doing something. While this might sound counterintuitive, human data has shown that this network is extremely important for cognition, imagery, memory, introspection, social cognition, and so on. And this network is the only one that coactivates with the hippocampus. And Now, two things are interesting here because they went a bit against my own result when I was a postdoc from 2004. So first off, it's really a two-way interaction, and actually the sharp waves were not even the most important part in it, as other oscillations showed stronger effects. So one thing I learned from this study was that in neuroscience you can't trust anyone. You can even trust yourself from 15 years ago, and the results you're mostly known for. So remaining enamored of your key discoveries might be convenient in the short term, but is an extremely dangerous attitude for yourself and your research field. So this work that I just presented is from Rafael Pedrosa, who is now in London. And you see that he mostly looked at sleeping mice. So Morganodran is now going beyond just looking at sleep, and she's testing how spatial memories and exploring environments affects global brain dynamics. So you probably got at this point that this needs to be done under this fancy microscope, and fancy microscope is as big as this table, so it's not something a mouse can easily carry. So the technology for now is getting there, but it's not quite there, that miniaturization. So we're stuck with the big fancy microscope, and then so the mouse is head fixed here, and what you do is you create a virtual environment, a virtual reality, and when the rat runs or the mouse runs, the environment moves with him, right? So this is Morgan's version that she set up in our group, which looks really great. So what does this all mean? So we still know very little to formulate a full-on theory, which is still quite difficult to do with what we know about the brain. So we have some simple ideas, which we wrote down in a review paper with Carola Keffler, Federico Stella, and Bruce McNaughton. In simple words, the cortex and the hippocampus together, not to speak about the rest of the brain, make not just two memory systems, but many memory systems. Some are small, and they involve just a little tiny place in the brain, like here. That would be responsible for different details in a scene. So that could be here, the microphone of the singer. Here could be some notes of music. Then you have slightly bigger networks that might record elements of increasing size and complexity, and then networks of the entire, representing the entire brain would actually correspond to very big association, perhaps the entire scene. So there's still much to flesh up, but this is a beginning which is supported by data. So by now, I gave you some ideas of some of the things I've done in the last 30 years, leaving out a lot of more work with great collaborators because of time. So let's finish this lecture by giving an idea of what my group and I are doing at the moment. So you have been hearing a lot about technologies in this lecture, and indeed neuroscience has been quite a lot in the last decades, perhaps even too much about methods, which are turning the immense complexity of the brain in something that is a slightly less of a black box. So as I mentioned, we're still a data-driven discipline. But things have changed a lot in these days. So when I started my journey, neurotechnology, we weren't using that term, was a craftsman matter. In order to prepare devices for tetrode recording in the McNaughton's lab, we were spending quality time in the mechanical workshop, and even postdocs had to learn the basic of machining. And in Paris, I learned actually to use watch repairman tools. But nowadays, we can do a lot more. So we have sensing technology that developed a lot, and now we can do chemical sensing, probing brain activity with magnetic fields, ultrasound, optics, and so on. So some of this sensing and the actuators can even be embedded in the neurons themselves using genetic technologies. So these advances cannot be the results of the neurophysiology lab bricolage, but are developed by a variety of specialists, electrical, optical engineers, geneticists, and so on. For their work, neuroscientists increasingly have to collaborate with neurotechnologists, and it's always a two-way exchange, let them understand your needs and understand what they can do. So this is a great thing about neuroscience. So it is probably one of the most interdisciplinary fields of all. And you cannot expect to become a specialist of everything. Perhaps you won't even be a specialist of anything. But you need to learn a lot of different scientific languages. You need to work in teams. You need to build teams. So that is what we did with the consortium Neurotech, which we founded with a group of Dutch colleagues a few years ago. And then with the Gravitation or Zwaartekracht project, the Dutch Brain Interfaces Initiative, or DBI2. So some of you may already have heard about devices like cochlear implants that can help some deaf people, and the promise of using brain implants to help blind people and people with a number of nervous system conditions. There is even the promise of boosting the cognitive abilities of healthy people with more or less invasive devices. So brain interfaces are becoming big businesses with big tech industries and Silicon Valley billionaires investing in it. Building brain-machine interfaces requires a very diverse expertise, as you probably figured out. So the Netherlands has a lot of researchers covering all the components, and with a group of them, which I have the honor of coordinating, we put up together the DBI2 consortium. And this is how we got one of the biggest subsidies in the Netherlands, now almost 1 year in operation. So it's a real powerhouse of neuroscientists, computational scientists, neurotechnologists. So building a new interface, especially something that you can implant in a person and maybe one day may allow paralyzed people to walk again, it's really takes a really long time. And that's a good thing, because besides the scientific work, you have to make sure that the device is really safe. Even at 10-year projects such as DBI2 is not enough for that. So what we are doing is to come up with a blueprint using computation and animal experiments for what a brain interface may look like in the future. So in fact, the current brain interfaces, you can say, on one side they are quite astonishing, on the other side they are quite limited. And that's often the case with early technologies, so they kind of look and feel a bit artificial. So we want to improve this with two key ideas. First, you've heard beforehand that different places in the brain, different structures talk to each other, but it's always a two-way dialogue. So a device that wants to integrate nicely in the brain should do the same thing. Listen to the brain and reply in the appropriate way. This is not completely new. It is called closed-loop stimulation, but it's easier said than done, especially if you want to do it at scale. So you have to read brain activity at the level of detail that I mentioned before. You need a computational algorithm that can process those data and calculate just the right stimulation pattern to shift the brain from its current state to the state that you want to reach. You need computer hardware that can make that computation in a very short time that you can measure in milliseconds, and you need very refined stimulator. So we have all that expertise in DBI2. The second point is in a way, even broader. So most neuroscience is done in the lab under very controlled conditions. This is a very good way to get reproducible results that can easily be interpreted. But at the same time, we want to understand how the brain works in the wild. With brain interfaces, we want to help patients in their homes on the street and not just in a lab or in a hospital room. And that changes things quite radically. There is a problem of validity, that is to say, it is unclear, sometimes it's not at all warranted, that our lab conditions and protocols are a reliable model of the real world, that the conclusions that we make can be extrapolated to real life. On the other hand, doing experiments under more realistic conditions, you need miniaturized, wearable implants, you need to track and analyze animal behavior. The analysis becomes a lot more difficult. But at the same time, it seems now to me, and increasingly many others, like a must. Again, with DBI2, we have brain power to make real progress in the field. So we have come to present days, and you see how that I was blessed with an exciting journey which brought me and my road fellows to many different exciting places. As far as I'm concerned, of course, the journey doesn't stop here, so where do we go next? So here are some initial ideas. So you have heard that the history of neuroscience of the last decades has been the history of methodological developments that have helped us getting increasingly more precise data. We went a long way, and you probably realize that I'm as much as a techno geek as anyone else, but honestly, I'm not sure anymore that we will make much more inroad in that way, that more data will lead us to more insight. The theoretical framework of neuroscience, this is my probably unpopular opinion, is and remains quite weak. So there is an elephant in the room of this lecture. So some of you may have realized that I never said even once that the brain processes information, that the brain is a computer, which is what many neuroscientists would tell you. Computational brain metaphor linking the biological brain to the brain-inspired AI is probably reaching its limits. Matching computational elements, computational primitives, to brain elements may give us some insight, but is ultimately a very difficult match. I say this after 20 years of looking at neural data and trying. I'm definitely not the first one or the only one saying this, but it is perhaps necessary to forget about the brain as a computer for a moment and start or go back to looking at the brain as a biological system, part of an organism with a phylogenetic history, the evolution, with an ontogenetic history, development, making feedback loop with the body, with other animals, and with the environment. So the neuroscientist and philosopher Umberto Maturana, an early critic of the computational brain paradigm, said in 1970 that, simplifying a bit, computation is in the eye of the beholder, so someone who's doing experiments and looking at the brain, but not so much in the brain intrinsically, and I increasingly subscribe to that view. A first step we're taking to go in that direction is to looking at brain dynamics under a different lens. Instead of looking at space, memories, and so on, we are looking at what happens when different animals communicate emotions between each other, moving away from cognition, information, and ultimately computation. I don't have the time here to explain much about it, but I took the gamble of shifting a large part of my group to this new research topic, and it's a gamble that I'm quite confident about for two reasons. The first is that we hired many great people to do this, and the second is that this is a project in collaboration with Valeria Gazzola and Christian Keizers in Amsterdam, who are luminaries in this field. So I hope that I will get a chance to tell you about the results of this new endeavor in the next lecture, which hopefully won't be my farewell lecture, and hopefully with some new ideas. So now we come to the thank you part, and doing this properly would take the entire hour. Perhaps that is what all inaugural lectures should do, because science is a collective effort, and there is very little that is just a personal achievement. People I collaborated with exchanged ideas, data, codes, stories, and whatnot. So we go into the hundreds through the years. So I thank my mentors, Daniel Amit, Alessandro Treves, Bruce McNaughton is here, Sidney Wiener. You see how shamelessly lucky I was. So Cathy Gotthard was technically not a mentor, but she's somehow responsible for me being a neuroscientist and stayed to this day as a sister in science. So even more important, I thank my mentees. Too many to name them all, but I really want to call out everyone in my current group in alphabetical order: Angela Zordan, Eric Kim, Davide Spalla, Francesca Bela, Guido Meyer, Jeroen Bos, Morgano Dren, Rotinda Bilek, Veronique Stockers. So we have 3 new PhD students who just started, so it feels a bit like a new group now. Federico Stella, now a PI by himself, but who remains a great friend and collaborator and a massive resource. My Nijmegen group of the last few years is really one of my biggest reasons for pride, not just for the science and the intellectual level of the group members, but for the group dynamics and the bond between us. It's one of those cases where you suspect that maybe you did something right, not just me, but everyone, and in particular your own boss, who is the man in control when it comes to experimental activity and a cornerstone of the whole operation. I would like to thank my colleagues in the Donder Center for Neuroscience and the DCN directors that succeeded themselves during my time, Paul Tiesinger, Richard van Wezel, Sean van Opsal, Eric Storkebaum. This place changed a lot in the 10 plus years I was here, and it has become a really exciting place to be. So I'm proud to be part of this story. So I thank the staff and management at CDL and TNU and the Animal Welfare Board. So everything we do in the lab depends on your often very difficult work, yes. So animal experimentation could be easier if the rules were a little less Byzantine. I thank the rats and mice that we use in our experiment. We always try to treat you well and to use as few as possible of you. I thank Radboud University and the Nijmegen Neuroscience community and the Donders Institute for being such an intellectual, inspiring, friendly, and pleasant place to work. I would also like to thank my colleagues in the DBI2 gravitation program. Again, too many to mention them all. It's been a very difficult work putting together this very diverse group during the pandemics and now getting out of the build-up phase with great help from our project manager, Suki Shin. Also an extremely exciting process. We'll do great things together. I thank my friends, our Amsterdam Italian community and friends who are here today. You are a lot of fun after our support network. I thank my parents for really making this possible. My mother was here, ciao mama. My father, who left us a few months ago. I thank my daughters, Irene and Sofia, for keeping up with crazy parents and for being the fantastic human being that they are. Much better than me. Most important thank you goes to my wife, Paula. So we came to the Netherlands 17 years ago as young scientists full of hope. We have 2 kids, raised them, managed to have 2 parallel academic careers in different fields and different institutions, and both getting to be professors. So this wouldn't have been possible without enormous amount of patience, mutual understanding, mutual advice, saving each other from the most blatant mistakes, working all the time for years, going through good and tough moments, teaching and learning from each other, and still it wasn't a given that this would have worked out, so it took luck as well. But what I'm mostly proud is that we did this together and we still love each other. And that's it.
Professor Battaglia, thank you very much for this inspiring inaugural lecture in which you took us along your own journey in the academic community. I hope it also inspires especially students to knock on doors of people they do not know that well, but they may expect some surprising thesis subjects from. You also took us along, and I think we had a great example on the first row of transfer of long-term memory from the hippocampus to the cortex. Okay, I thank you also for leading us into several aspects of the brain, and especially for your inspiring last part. Where you told us not to compare the brain to a computer. And in your endeavors to translate this also to a much more biological setting for your animal models. And I wish you all the best in that endeavor. Then I would like to thank all those present here and through the live stream. I also think there's people joining through the live stream. It was very good to have you here, and I think I hope you just enjoyed the lecture as much as I did. I certainly did. And then there's some practical issues. So after this ceremony will be closed, the cortege will leave the room, followed by everybody else. Drinks and snacks will be available on the ground floor. Don't form a very long line. You will see that the cortege usually forms the line, but just walk on, get a snack and a drink, and then there will be ample opportunity to congratulate Professor Battaglia. And then lastly, of course, I would like to thank Professor Battaglia and congratulate him and his family on accepting to be a professor with us. And not in the least, I would like to congratulate us for having Professor Battaglia amidst us. Then we close the ceremony, and I would like to ask you to please rise. And according to the customs of this university, we close by the words, Gratias tibi agimus, omnipotens Deus, pro omnibus beneficius tuis, qui vivis regnas per omnia saecula saeculorum.